Hi, welcome to Neuroverse
A podcast hosted by Carolina.
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on science misinformation. I was inspired to do this episode because a friend of mine sent me an article. It was a clickbait article about animal human chimeras. And this article referenced a Joe Rogan clip where
What a good reference.
Yes.
So reliable.
So Joe Rogan and Alex Jones were talking about human animal chimeras, and they went on to these what I can only describe as
False.
false and just very much out there conspiracy theories, and very much fear mongering as well about the research done around human animal chimeras. So I thought it's very important to set straight science misinformation, and I picked this topic to clarify some things. So to start off with, a chimera is an organism that is composed of cells that are distinct from one another. So it carries two different genomes. This happens naturally in different species of flowers, and stem cell research with induced pluripotent cells as well as embryonic stem cells has been exploring this idea of human animal chimeras or interspecies chimeras for therapeutic reasons, such as either cell transplants as well as organ transplants, and many others that I'll discuss further on.
So, what is the use of stem cells to create these?
So, the way it happens is, and I'm going to reference a really foundational paper by Toshiro Kobayashi and Hiromitsu Nukayuchi, where in 2010 they were able to grow mouse that had a rat pancreas. So this was done by taking the mouse blastocyst, so the very early stage of an embryo where the cells are undefined, and they then deleted the gene in the mouse that it requires to develop a pancreas. This gene is the PDX1. So by deleting this gene, the mouse wouldn't be able to grow a pancreas by itself. So what they then did is they took induced pluripotent stem cells from rats. Induced pluripotent stem cells are cells that have been reversed back into their undefined stage or unspecified stage with transcription factors. And by introducing these cells into the blastocyst, by introducing the rat cells into the mouse blastocyst that lacked this gene, it covered the biological gap that the mouse required to produce a pancreas. So by introducing cells that didn't have the deletion, these cells naturally grew into pancreatic cells and filled the gap that this mouse required to survive. And so then it survived, and it had a normal functioning pancreas and was able to produce insulin.
So, just to clarify, the cells that then turned into pancreatic cells in this mouse came from a rat, but they were induced pluripotent stem cells.
Yes.
So they were in a state in which they had a lot of potential to become various different identities. So the fact that they came from a rat is cool, but it's not that significant. Like, it could have come from any other animal.
Yeah. The reason why they used rats and mice is because evolutionarily they're very close to each other, and that helps with the survival of the organism. So this is to sum up some of the more foundational research done with animal chimeras. And the point that I want to make is that it wasn't some morphed mouse rat. It was a mouse that had a rat, quote unquote, pancreas. So,
but barely, because it wasn't like they took the pancreas from the rat and put it, like,
exactly,
it developed into pancreas within the mouse.
Yeah. And so the DNA wasn't mixed or fused or anything like that. And in this article, in this clickbait article, they had a lot of weird human pig images of just some crazy morphed pictures.
Probably Photoshop.
Yeah, but my friend didn't know that this was Photoshop.
Well, yeah,
she was concerned that these were real images and this research was really happening. And so I explained to her that that's impossible, because in chimeras, all you're doing is adding a few cells that then grow into an organ.
Yeah.
if they do even develop to grow into an organ, and the DNA doesn't morph.
Yeah, and how is this then used with human stuff? So you gave an example of mouse and rat, but
Yeah.
how are human animal chimeras? What are some examples of human animal chimeras then?
So the most recent and successful human animal chimera was done with macaque embryos, and this was done in 2019 with a paper published in 2021. And essentially it's the same principle. So they have a macaque blastocyst, so the embryos with undefined cells, and then they tried to add a few human cells in a petri dish. So they didn't
Emphasis on in a petri dish.
exactly. So they didn't implant the blastocyst anywhere. This is all done in this specific experiment, and the ones that are done with human animal chimeras is done in a petri dish where the blastocyst is a few days old, and the maximum days that the cells were able to survive was only 19 days. So it didn't even have time to develop into like a full stage animal or anything like that.
Just to challenge you though, isn't the goal to eventually make it so that it would be able to develop into a full organ or animal?
Definitely. The goal is to then, because the goal of this research is to help with therapeutic needs, and such as the need for an organ transplant. In the US alone, there's 100,000 people on the waiting list for an organ transplant. So the goal is that. However, the fact that there are articles out there insinuating that blastocysts have been implanted in animals, and insinuating that there are full-term pregnancies being taken place, and insinuating all sorts of these things. I think the point of this podcast is to convey the true information, and that this is the reality of where the research is at. We are very much in the early days. We have been successful with other interspecies such as rat and mice, but so far human animals, it's still very much in the early days. And there are doubts in relation to this research, which I think are important to address because we're trying to be transparent here. So some of the doubts is that when inserting these human cells, that they can differentiate into brain cells, and raising issues related to consciousness. The second doubt is where human gametes may develop inside the animal. And the third doubt is weird modifications that the animal may have a human aspect, such as skin type or facial structure. Now, the original pioneers of this type of research, Toshiro Kobayashi and Hiromitsu Nakauchi, addressed these doubts in their paper Revisiting the Flight of Icarus, Making Human Organs from PSCs with Large Animal Chimeras. And as a little side note, I really like it when papers are a bit creative with their names. And,
definitely, yeah.
In this case, the Flight of Icarus is about Icarus, who was trying to flee away from danger. So to fly, he employed wings that aren't part of his normal human form. So to address these doubts, they state that from their previous interspecies chimera experiments, they have never seen these barrier limits crossed in terms of a morphed rat mouse, in terms of the concerns of a humanized pig. And the number of cells introduced are very small anyway, so it wouldn't even be able to be half human, half pig in that sense. They also say that unless the cellular niche is provided, in other words, unless like brain cells are provided, brain cells won't be developed. And so all we need to do is simply not introduce brain cells, or not introduce a genetic mutation that deletes the host's brain cell making capability. Yeah. Yes, exactly. So as long as we don't introduce the cells, it won't develop. So we have control in that sense. Also, with the concern around gametes, they have even greater confidence. However, they are developing targeted organ generation methods, which through the transcription factor MIXL1, they were able to induce differentiation of pluripotent stem cells, specifically towards an endodermal lineage. And they are also introducing suicide genes that under neural or germ cell specific promoters eliminate the formation of human pluripotent cells in the host. So not only has none of the concerns that are currently present around human animal chimeras happened in previous research with other animals, but we are also developing mechanisms that will simply not enable them to happen, such as these suicide genes and a specific endodermal lineage target.
Yeah, it's obvious that I think scientists understand that the work they do needs to have a significance. So the work with chimeras that they're going to be doing is for the particular use of these chimeras for, as you said, like organ development, for transplantation and positive outcomes. And then sometimes science can be miscommunicated because non-scientists forget that there's so many regulations in place as well, and that if you aren't doing science correctly, you'll tend to be frowned upon and not like be rejected by the scientific community. And so it's just not even like science isn't for fun, you know?
Yeah.
Especially for something clinical like this. It's for the clinical use.
Yeah.
And so, yeah, it makes sense that they have to be thoughtful about what they're doing and make sure that they're doing it correctly.
Yeah. And it's not only organ transplants. Other examples of therapeutic uses include hepatocytes for inherited metabolic disorders. And like you said, there's so much regulation around scientific research. You know, whenever we have a research idea we want to explore, we need to apply for grants, and that can take years, and we need funding for it. And funding in science is incredibly scarce. You know, it's not like these underground labs that have millions of cash and are just doing these like crazy research. No, that's
the dream, but
yeah,
it doesn't exist, unfortunately.
Yeah, this research is for a purpose. It's highly regulated, and funding is incredibly limited. So
yeah. But also, I agree that there can be an ethical standpoint where you are against this, or like against using animals in research, and I think that's a different subject. But just for like science misinformation or miscommunication, like there is no point in forcing people to, or like encouraging people to come up with conspiracy theories of like scientists having alternative motives, and like literally, what would be the interest of anybody to create a human pig?
Yeah.
Like, then what would you do then?
Yeah. Like, I completely agree with what you said about how, you know, in terms of ethical and moral opinions, you know, it's up to the individual. However, yeah, the purpose of this episode is about science misinformation, and there are a lot of clickbait articles out there. And while this can be a controversial topic, it's important that, you know, we know the full information, we know the doubts, we know the reality of the research. And the reality of the research is, so far we only have at best a bunch of cells, and they're not even full animals. So any images or any clickbait articles you come across are fabricated.
Yeah. So I think it's interesting to explore, though, like why and where people are getting these ideas from. And so before this episode, Carolina shared with me the YouTube clip of the Joe Rogan show where Alex Jones was describing the absurdity of human-animal hybrids. And so this was before we discovered that Alex Jones is an American conspiracy theorist, as his Wikipedia description. But he presented some evidence from a article saying, quote unquote, MIT Technology Review estimates that about 20 pregnancies of pig-human or sheep-human chimeras have been established during the last 12 months in the US. And so we came to the realization that the main problem with this statement is the use of the word pregnancies, which is false, because Carolina explained to me that this wasn't actually a pregnancy.
Yes, it wasn't a pregnancy. It was a blastocyst in a petri dish. And
again, emphasis on petri dish.
Exactly. And the word pregnancy almost it insinuates that these cells were implanted, but they haven't been implanted,
and that like an animal went through giving birth to a human, which sounds traumatizing.
Yeah. So and this is like, that didn't happen.
It didn't happen, and it's clearly like sensationalized language.
Yeah.
Just to get people intrigued and interested and emotionally aroused or whatever.
Yeah.
Yeah.
And on that note, Martin Bobrow wrote a comprehensive report into the subject about mixing or in the introduction of human DNA sequence into animals. So there are reports and there are papers out there that are completely open source and up to the individual to read. Science tries as much as it can to be open access, and the reports are completely honest and just reports of the information that we have. And articles like this, they choose their words carefully, and yeah.
Yeah, I think with science communication, it's difficult because you're always trying to simplify language for the layperson, of course. And sometimes this can naturally cause miscommunication, but in a case like this, I think it's so easy to choose language to purposefully fool people. Like, you can also say in a petri dish, and or like, you know, in a dish,
Or
or
outside
in the
of
lab,
the human,
or
yeah,
anything,
or outside of the animal.
Yes, exactly. So there are other options,
but they chose the word pregnancy, which
yeah.
has so many, ethical attributions to it.
Yeah.
So we understand that this topic may be quite controversial, and people have different opinions on it, and it's a difficult topic to talk about because it's very new technology as well. But we hope that this was informative and that it clarified any doubts, and also hopefully made you a bit more aware of science misinformation happening in the media, especially sensationalized stuff. And even though I personally believe that big platforms should have some responsibility about fact checking and make sure that they're only propagating true information, that's not the case. And so I encourage you to double check the facts whenever you think they may perhaps be a bit sensationalized.
Thank you for listening.